now, and I think we'll kick off, and hopefully any others will catch up later. So welcome to this first of the super interest group sessions for Genomics and AI, organized in conjunction with the Turing Institute and hosted by the Genomic AI Network from the NHS. Nice to meet you all. My name is James. I'm the director of the network, and I'm joined today by a couple of colleagues. Unfortunately, Alex, our clinical lead, who is due to be our regular host, is actually away at the moment, which is unfortunate timing for our first one. But just to give you some kind of background for this, we envisioned this session as sort of a regular sort of meeting point for the genomics community to come together and discuss, particular challenges and opportunities from AI. And the kind of format we're kind of, going to go for is each session we'll kind of bring in a guest speaker to talk about a particular aspect of AI, and then we'll make sure we've got plenty of time for discussion afterwards. So I'm going to hand over to my colleague Wes, who's going to run through the format of the day before actually kind of starting the session itself.
Wonderful, thanks, James. Good afternoon, everyone. My name is Wesley Pigg. I am the project manager for the Genomic AI Network. So just in terms of the agenda for today, we have Francisco, who's the director of Bioinformatics at Genomics England, will be doing a talk on AI-enabled genomic medicine at Genomics England. Following that, there'll be about a 20-minute session, and then following that session, it's essentially just an open slot for questions, and we welcome any questions. Please do use the chat, etc. The aim of these sessions obviously very much is for that collaborative discussion. So we very much look forward to that, as well as Francisco's talk. So that's basically the agenda, nice and simple, which is great. Just in terms of quick housekeeping before we kick off, the meeting will be recorded, and the intention is that this will likely go on our website as a resource for people. If you could please just make sure your mics are on mute, and then again, if you can keep your questions to the question session, that would be fantastic. We can't turn off the ability to raise your hand. So if you can just avoid that, just to help us with keeping the keeping time for the session. And then obviously that will help us have plenty of time for the actual question slot. But I believe that's all in terms of housekeeping. So I think rather than having a delay, I'll hand over to Francisco.
Thank you, Wes. Good afternoon, everyone. I am Francisco Azuaje, Director of Bioinformatics at Genomics England. And today I'm going to share with you how Genomics England is developing AI to advance genomic medicine. I will walk you through some examples that illustrate our current journey and how these technologies are transforming the way we do research and support patient care. We'll begin with an introduction to Genomics England, and then I will explore some examples of AI applications across the genomic medicine workflow. And to conclude, I'll share some learning lessons and takeaways. Genomics England. First of all, an introduction for those who don't know us yet. The infinity loop that you see here is our best representation of what we do and how we do it. At Genomics England, our mission is to enable better healthcare outcomes for our patients and accelerate genomic research. The data originating from our healthcare mission enables further research, and the outcomes of such research feed back into clinical care, including new diagnostic insights. At the same time, the center of this infinity loop now offers an opportunity to generate evidence for the acceleration of innovation at the intersection of healthcare and research in sequencing, analytics, tooling, and AI. Before discussing some of our AI-powered applications, let's briefly look at the healthcare AI landscape in the UK, where we see encouraging trends in public acceptance of AI in healthcare. For example, a recent survey shows that more than half of the UK public supports using AI for patient care, while NHS staff support is actually higher at 76%. Interestingly, this support increases when talking about the AI applications for administrative applications. And this growing support, this growing public confidence provides both a strong foundation for responsible AI implementation in healthcare and opportunities to continue developing that trust with healthcare professionals leading the way in recognizing the potential of AI for healthcare. Based on recent findings from the Genomic AI Network, this diagram shows the current distribution of AI tools in the NHS genomic pathway. As you can see, the dominant focus currently is on variant interpretation at more than 73%. As you know, variant interpretation is a critical step in defining which genetic variants might cause a condition. Patient identification and pharmacogenomics each account for about 9%, while clinical decision support remains relatively underdeveloped. And looking at developers, we can see that Illumina leads the space, followed by different companies and academic institutions. So this picture highlights both progress made to date, but also opportunities to develop AI solutions for different areas of the genomic medicine pipeline. Now, imagine a patient's genomic data beginning its journey through the healthcare system. At each step, there are opportunities for AI to enhance our understanding and improve patient care. Beginning with core data processing, we handle the task of genomic and clinical data acquisition, applying rigorous quality control. Then this fits into deeper sequence analysis where algorithms, including AI algorithms, help identify genetic variants. Moving around the cycle, we see how multiomics and multimodal data integration helps build a more complete picture of each patient's condition. Then more recently, large AI models are becoming increasingly important in understanding complex genomic patterns. This understanding then flows into clinical decision support, treatment planning, ultimately aiming to provide better care for patients. So at Genomics England, we are focusing on a number of these areas with different levels of efforts and also different approaches. And I will shortly walk you through specific examples of how we are implementing AI in some of these areas. In terms of AI, our mission Genomics England centers on accelerating genomic medicine, and the responsible application of AI here plays a key role. So I want to emphasize here 3 core principles that are guiding our work at Genomics England. First, we ensure that all AI solutions are developed with robust safety protocols and validation processes, in particular data privacy. Second, every implementation, even if we refer to POCs or early discovery, must demonstrate clear benefits for patients, a clear connection with a clinical problem, including improvements in patient care, patient outcomes. And third, we design our systems to augment, not replace, human expertise. This human AI collaboration is anyway essential for delivering better healthcare. At Genomics England, our main approach can be briefly summarize here. And this is based on the integration of multiple data types using different pipelines with AI incorporated at different levels of the pipeline. So we begin with molecular level genomic data and other data types, including clinical imaging, medical text, to find associations between them. Typically, this data undergoes two parallel processing paths. The first one is an expert engineer feature approach where features are based on domain knowledge. And the second is a machine learning based feature extraction for both structured and unstructured data. Then these features fit into different unimodal and multimodal machine learning systems for either healthcare or research applications. And these systems produce different outputs and insights from quality control and variant classification to diagnostic support, biomarker discovery, and knowledge synthesis and curation. Here is our first concrete example of AI in action, detecting degraded tumor samples using AI. This example shows AI's impact on a crucial clinical problem, a crucial component of our bioinformatics pipeline. Within our genomic medicine pipeline, there was a need to improve the identification of poor quality tumor samples or degraded tumor samples. Before there was a limited detection rate that has an impact on the efficiency and the quality of patient care, because degraded samples can lead to inefficiencies and also to inaccurate results. So our team trained a machine learning model on 700 expert label samples, poor quality, good quality samples, achieving remarkable results. For example, we have reduced the false negative rate from more than 90% to just 0.5% at the moment. Also, by adding just a modest increase in false positive. And this model has been successfully deployed in our production environment and is now significantly improving sample quality assessment reliability, which is essential for accurate downstream analysis. Moving to our second example, we have tackled another crucial challenge, which is evaluating the performance of tools that predict whether genetic variants are likely to cause disease. So here, using our high-quality curated clinical data, we evaluated 27 prediction algorithms for detecting harmful genomic variants against clinically validated variants. Through a rigorous three-step evaluation process with different performance criteria that you see here on the right, we identify top performing models, including meta-RNN, BaseDel, and others, that achieve an optimal balance of predictive power and low error rates. Although we didn't find a universal solution that is good at all cases, including the prediction, including error rates and predictive performance, we found that at the specific thresholds, for example, the top 3 tools correctly identify benign variants 83% of the time, and pathogenic variants 96% of the time. So this benchmarking is relevant not only for enabling further research, but also it can help clinicians select the most reliable tools for their diagnostic workflows. Now, let's turn to another exciting project that demonstrates how AI can transform our literature review process. So finding relevant disease-causing variants is a complex challenge. It's time-consuming. Just to give you a sense of the magnitude of the challenge, from a patient 6.5 million genetic variants, we need to prioritize just around 10 variants relevant to their condition. And gene panels are essential for this filtering. And gene panels are annotated using different sources of knowledge, including the literature. And keeping these panels updated with the latest research is challenging. So to support our biocurators to tackle this challenge, we develop a Gen AI-based system that has processed more than 200,000 research articles to identify new gene disease connections. So far, the results have been very promising, including, for example, 40 potential gene updates within one of the most used diagnostic panels, also new associations, and other associations that may be relevant across panels. So this literature, this AI-powered literature analysis ensures not only that our diagnostic panels incorporate the most current scientific knowledge, but also allows our scientific curators to process information much faster. Now, let's explore how we are using AI foundation models to generate genomic embeddings and support multiple downstream applications in cancer research. So as you probably know, foundation models represent the next step in the evolution of genomic AI. We have implemented a cloud-based pipeline using the nucleotide transformer to generate meaningful genomic representations using different approaches across patient cohorts. And these embeddings or these meaningful representations are very important because they can be used as the inputs for further downstream applications. And for example, when you visualize these embeddings, you can see at the very beginning how they naturally cluster by gene type, how they cluster on the basis of other biologically meaningful groups. And that demonstrates the potential relevance to create compact, informative representations of DNA sequences that can be later used to implement other AI prediction models. This research is actually part of an ongoing collaboration of Genomics England with InstaDeep. We are applying the nucleotide transformer and other models to analyze the sequences, the DNA sequences from thousands of cancer patients. This diagram here illustrates a high-level view of our foundation model pipeline. I like to emphasize that this isn't just about a machine learning POC or a pilot. It's more than that. It's about creating and investigating more meaningful representations of genomic data that can help us better understand disease mechanisms, patient outcomes. And as you can see here, we start with patient-derived data. We apply various training approaches, including self-supervised learning, using foundation models, and then use the resulting models, the resulting embeddings for multiple downstream applications, from patient classification to treatment response prediction. Beyond genomics, we are also applying AI to support a number of pathology imaging analysis tasks. And here is an example. We have an extensive data set of tumor histopathology slides, and before sharing that with the community, before using them as inputs to enable machine learning, these data sets require proper curation, augmentation, and among those tasks, the challenge of correctly classifying tumor versus normal tissue is essential. It's essential for improving the usability of those data sets. It's essential to reduce the noise included in the data set. And this in turn can enable more accurate predictions downstream, predictions of clinical outcomes such as survival, biomarker status, patient clustering. So we have developed an AI system trained on 800 expert label slides that achieve 97% accuracy on a test data set. And this system not only classifies entire pathology slides, but also generates detailed tumor segmentation masks, which are useful for precisely localizing the tumor tissue within the images. And then this can be seen as a way to explain the prediction and facilitate the verification of a prediction by a human expert. And this system has been applied to our collection of more than 12,000 colorectal cancer slides, enabling more precise analysis and facilitating integration with genomic data. As I approach the conclusion of my talk, I'd like to highlight a few takeaways that we have learned about the implementation of AI solutions in genomic research. So I like to highlight that success in AI-enabled genomics research rests on three fundamental pillars that you see here. First, solid foundations, including technical foundations, clear clinical needs identified, that will allow us to build systems that can adapt and scale with healthcare needs. So we are not only developing, we shouldn't be developing tools only for 2025, for example, but we are developing tools that can be adapted and expanded. Second, trust. This is essential to Genomics England mission. This means developing transparent, ethical systems that healthcare professionals and participants can rely on. And third, collaboration. As you know, no single organization can do this alone, but also within your organization, you need these solid interconnections, these collaborations. And the key message here is that these pillars are interdependent. The key message is that technical excellence, for example, without ethical considerations and effective management of collaborations won't succeed, just as great partnerships will not help if the foundations are not solid. To summarize the key messages from today's presentation, first, AI is transforming genomic medicine with practical applications throughout the workflow, and I illustrated a few examples. Second, multimodal, multi-omics approaches that combine different types of omics data with clinical information and other data types enable more powerful insights. Research insights, but also is moving us toward truly personalized medicine. And finally, successful AI implementations require robust data infrastructure, robust collaborations, transparent ethical systems. And that's yeah, for me, I want to thank our incredible teams at Genomics England and partner teams who have made this work possible. In particular, a massive kudos to our ML engineers at Gel, Andrea, Stanley, and Sam. And thank you for your attention, and I'm happy to take questions or comments.
Francisco, that was a really interesting talk and some really useful examples you highlighted there. If you don't mind, I'll ask the first question actually. So you highlighted at the end some of the kind of the core principles you think you need to have in place to make AI a success. I'm curious to know from your point of view, what's the biggest single blocker at the moment that's actually kind of preventing us from kind of moving from the kind of useful research examples you presented into actual kind of clinical practice?
Yes, into clinical practice and in research actually as well. I think the biggest blocker for me would be access to high quality, useful data. And here we are not just talking about the volumes of the data sets or even the clinical relevance of the data set, which is essential, of course, but talking about a condition in which the data sets are sufficiently informative, that can be connected to other data sets, that can be accessed for ML purposes. And that means more than volume and access. It means quality. It means the right level of annotations. So it's about having the right data and also having the capacity to make this data useful. So that's a key requirement for me. I think people talk a lot about quality, but I think it's all these components about accessibility, quality, and usability that goes beyond errors and noise. I think the other obstacle for me is to find ways with the community, including the NHS and the research community, to have systematic approaches to evaluate tools and have a clear pathway towards adoption and deployment. So if I have to pick two, these are the two main concerns for me.
Just quickly on that first point you made on the kind of necessity of having kind of good data, had Gel kind of already been mindful of that? You know, how much work has Gel done on top of what you'd already had in place to kind of enable AI tools, or was your data already in a kind of good state?
I think on the one hand, we have the advantage of having access to a unique, high quality library of data. I think that the level of maturity for ML development is heterogeneous across data sets. So you may have, for example, genomic data. You might say this is not just the largest data set, is excellent in terms of annotation, links to clinical data and so on. Then you have other data sets, including clinical imaging, that are more recent, and then the level of usability, accessibility is different. So it's perhaps behind. But at the same time, regardless of this heterogeneity of data readiness, Genomics England right now is in a better position than a few years ago. I think also across the company, there is an understanding that ML requires more than just access to data. It's all these factors to make the data useful and reliable. So I would say this is part of a journey. I think that we cannot say that for all data sets, for all uses across the company, we are at the same level, but we are progressing.
Okay, that's great. Thanks so much. Let's move on then to some questions in the room. I can see we've got a few hands up. Shall we start with Christoph?
Oh, thank you. Yes. So, first of all, great talk. Thank you very much for that. So, I'm Christoph Podrewowicz. I'm Professor of Bioinformatics at the University of Bradford, but I'm also Elixir Human Copy Number Variance Community co-lead. So I actually want to make a question. I've got a couple of the questions, but I want to maybe make some comments about what Francisco just said. I think you've been talking about the FAIR. So FAIR principles, right? Yeah. Or FAIR data, FAIR metadata. So the way how you describe the making sure that data are findable, reusable. And also in terms of, I share your concern or your about making sure that we've got a good community of which will allow us to test the tools, but also test the data and introduce good practice. And I think this is something where Elixir can be very useful because we are dealing with the large number of the infrastructure, right? Also infrastructure tools related to the different types of the genomics and also got access to data. So I think that that's something very useful if you will permit, right, the community to engage. But my actual question is about the way how to engage, because you mentioned the Illumina as one of the major software provider. But I'm just wondering what would be the mode of the engagement, for example, for the researcher who are working very well at the local level with the different NHS trust partners in terms of the implementing or AI solution, right, or performing research using AI to the different mode of the data. Is there any way that you can offer the support and any type of the programs? Is that something which you are looking for as well?
Well, just 2 points on that. One, the Genomic AI Network is a key player already, and James and Wes can tell us more. But I think that's part of those community-driven efforts to not only to build community, but also to support the adoption and the deployment of AI technologies. Now, from a Genomics England perspective, there are programs that we have at Genomics England to support not only access to our national library, genomics library, but also to support the collaborations. And we have, for example, a research network, and there is within that research network you have different interest groups, and there is one focusing on machine learning, and these groups are driven by the academic community. So that's something I'd like to recommend to you and all the participants to have a look here at if you go to the Genomics England website, you will see that. Also, we have a program for industry that supports industry partners anywhere. Actually, it doesn't have to be the UK, to have access to our data, to tap into our networks also, and you have different types of collaborations.
Yeah, thank you. Thanks for that. Thanks.
Thank you.
Thanks so much. Let's move on to our next question then. It looks like it's Dr. Akshay Kumar.
Hi, thank you, James, and thank you for the wonderful talk. I'm a consultant neuroradiologist in a university hospital, Coventry, and I'm also co-founder of a radiogenomics company. So basically, we have an innovation which predicts the mutation of brain cancer using only MRI scan. It has an EI and other things. But when the main thing which we are struggling currently is that we don't have a robust data set, as you were saying, for the cancer specific relation, like you have for in US, like tumor cancer imaging archives and stuff like that. So my question is, are we thinking about making such kind of platform in NHS, which would be useful in long term to be conductive for research and innovation, and but also actually to just to have one stop place where researchers could collaborate. And because now I feel the research are very siloed in our NHS, because for example, some university would be doing something, but it may not be as much collaborative as it could have been. So
okay, so as you probably know, a key priority for the UK is to enhance the way healthcare data is shared and interconnected. So this is a longer term ambition, and many things are happening at the level of Genomics England. As I said before, we are already in a capacity to to support researchers from academia and industry to access high quality annotated data sets. As I said before, currently genomics data and clinical data are probably at the highest level of readiness for machine learning, for example, but we are gradually advancing towards multimodality, multiomics, and the goal is that those we will continue enriching, enhancing those existing data sets to include these modalities. This will be an incremental process because we want to ensure not only the highest standards in data privacy, but also ensuring that we have the right level of maturity and quality to enable research. So it's part of the goal. At the same time, I agree with you that there is still a need for providing maybe subsets of well-curated data for specific domains, for example, particular types of cancer or pan-cancer, that can be used to benchmark AI models and support innovation challenges, support research of all kinds. And I like to see that also coming from Genomics England. I think we are advancing. We are not there yet. And as I said before, the UK in general, I think, we have an opportunity here. And with the Genomic AI Network, we will be also in a good position to support that.
Thanks very much. Let's go over to Ajub.
Thank you. Thank you, Francisco. So I'm a co-founder of a AI health tech looking at automating meta-analysis through AI, open source large language models. But the program that you just mentioned earlier, where you were looking at 200,000 journal research studies, my question was, how are you able to parse and extract the data from all the different studies, from the different PubMed and other platforms? There's always a problem with the paywall issues to be able to extract data. Are you using open source large language models as well, or are you using your own models to be able to extract and systematically review and perform meta-analyses on these data sets?
Yeah, thank you. So as part of this project, we began with a focus on a collection of articles that was a plus journals. So after that, when we completed that POC, we expanded that to cover PMC Europe. So basically now we are relying on open access journals available through PMC Europe. So that's that has been useful to demonstrate the value, but also, of course, we recognize that this is a limiting factor. And you mentioned some of the obstacles. In terms of models, we are not producing our own models. We are in close collaboration with AWS. We are using Claude models. And our contribution has been mainly focused on the prompt engineering aspects of the project in close collaboration with biocurators. And we have these feedback loops of prediction generation, assessment, verification. So we are relying on this existing large language models. At the beginning, we performed some analysis using other ones, but right now our focus is on Claude, the family of Claude models through AWS.
Right. Thank you.
Okay.
Let's go over to Brian.
Hello, I'm Brian Schilder. I'm a researcher at Cold Spring Harbor Laboratory in the US. I was really interested that you guys are working with InstaDeep using nucleotide transformer for the patient embeddings. And I was curious about what specific disease indications you're using it for. I know you mentioned pan-cancer. And then maybe a little bit more on the details of how you're using these embeddings. Are you sort of training a secondary model with these embeddings to learn across different disease indications, or are you kind of creating disease-specific models, even like, within cancer or outside of cancer? And yeah, and some of the more the details of exactly how you are incorporating that genetic variation, whether you're actually making full personalized sequences or just doing sort of injection with like one at a time variants.
Thank you for the question. So first of all, with InstaDeep, we are evaluating also different baseline models using classic machine learning. So that's something to consider as well. And we are doing this with a focus on cancer. And here we are talking about applications such as predicting the origin of tumors of unknown primary. For example, clustering pan-cancer cohorts, subtyping, also some explorations involving the prediction of responses to treatment, such as PAR inhibitors. So this is something we have been doing with classic machine learning models as a baseline. Now, in the context of the nuclear transformer, we want to investigate how the embeddings coming from the nuclear transformer, the Polish version, but also a new version trained on Genomics England data, can support different downstream applications, including the ones that I mentioned before. So it's more like an exploratory analysis with a focus on cancer, and it's still early stage. And I think that in the near future, if I have to select two promising applications, on the one hand, the prediction of tumors of unknown primary, prediction of treatment outcomes, and also room for exploratory analysis, discovering subtypes of cancers. Here, the approach that we are using is we are focusing on whole genome sequencing data, but of course we are not in a capacity, as you know, to use the whole genome. So our initial approach is to focus on a filtering approach. First of all, according to the application task, we use domain expertise to select gene sets with clinical relevance, and where we know that variants in those areas, in those regions, have an impact. So that's our first approach, and down later we should be able to explore more less biased approaches. But this is a high-level view. Hopefully it's clear now.
Yeah, that's great. I mean, I'd be definitely interested in talking more because we're trying to do a lot of the same stuff in my lab, and taking a number of different approaches, including DNA-only language models, as well as specialist models, like protein language models. And we're in the process of actually getting access to 100,000 genomes. Used to have access,
wow.
But then I moved here, and we have to start over.
Oh, okay. But hopefully it's going well, and yeah, feel free to reach out, Brian, if you need.
Great.
It would be
Thanks.
great to catch up. Yeah. Thank you.
Absolutely.
Okay, I can't see any more hands up for now, so let's move over to some of the questions that were asked earlier while Francisco was speaking. First question comes from Belma, and this is, did you test alpha missense among your AI prediction tools? And if yes, how did it perform?
Yes, we did. It was not at the top five in terms of the overall measures of performance. But there is no significant difference, between alpha missense and the top three that I mentioned. I think the key message here is that some tools are good at predicting particular types of variants. It's very context dependent. But yes, absolutely. We tested alpha missense. It's a top performer. It's a top competitor, but it didn't come up as the universal, the best solution for all cases across all kinds of variants.
Okay, Next question is from Christoph again, and this was, do you use any workflow management systems in your analysis?
We are here tapping into data that have been already have been pre-processed by our bioinformatics workflows. So we are not reproducing the bioinformatics workflow, which is something that our bioinformatics pipeline implements different ways to orchestrate those pipelines. So we are using basically the data coming that is available in our research environment. And so in terms of workflow management, for ML, we are working with the tooling provided by SageMaker to ensure that we have pipelines and we manage experiments and workflows. But this is in the context of ML. Hopefully that answers your question.
Yes, maybe I can just jump in. Yes, I was asking about whatever you are implementing, and a part of ML analysis also using the workflow management systems, right? Yeah. So thank you.
Yeah. Thank
Okay.
you.
And actually, Christoph, while we've got you on screen, I can see you've asked another question in the chat. You asked about health data research, and if I guess Francisco has been looking at that in terms of other types of data other than genomics data.
Yeah, so that was mainly just to maybe point out, right? Yeah, because I think Francisco gave a very good answer, right? Yeah, but I know that also Health Data Research UK, right? Yeah, because they involved the multiple NHS trusts, right? Yeah, they can provide access to the other types of the data. I think there was a question about the types of the digital pathology data, right? Yeah, or cancer-related pathologies, images, right? Yeah, whatever there is any possible access within the UK domain. I think Health Data Research UK is one of your partners probably, which can facilitate that.
So for these use cases that I illustrated, we don't have any particular partnership in that area, but of course we are tapping into all the data set coming through us, and in the case of imaging, this is part of an initiative that involves the UK network for imaging, pathology imaging. But apart from these data types that are linked to our genomic data sets, we haven't explored additional health data sets.
And then the last few questions, I think some of these have already been answered, relate to your LLM-powered use case. How have the articles been processed to create gene-disease relationships, and was there human supervision? I'm not sure if you answered that one or not yet, Francisco.
So we are basically using a simple RAG approach to incorporate chunks of text coming from the articles and containing relevant information for the query, and using that as inputs to the LLM to not only identify the gene disease associations, but also the supporting evidence. So we have this filtering process of preparation of articles that we know are relevant for a particular condition and relevant for a particular gene. And then we perform that RAG approach to facilitate the processing by LLM. In terms of verification and evaluation, so we not only the prompt engineering, but also the analysis of prediction, but also the analysis of predictions are conducted in close partnership with a biocuration team. So initially, at the very beginning, we also have a gold standard of good predictions and bad predictions to validate models. Right now, what we are doing is a prospective evaluation that we produce batches of predictions on a regular basis, and these summaries of predictions with the supporting evidence are provided, are forwarded to the biocurators who inspect them and analyze the relevance of those predictions.
Okay, thanks. Just one follow-up question for me, actually, on the same topic. I'm curious to know if the fact that obviously, like all large language models, that Claude, there's various versions of the Claude models, and they change quite frequently. And if you're using kind of APIs, you're kind of somewhat at the whim of the kind of the companies as to whether they're going to keep these models available. Have you switched models at all, or has this all been based on one specific model? And do you see any problems with this, the need to kind of switch between models in the future?
Absolutely. I think that we all should be designing for vendor agnostic applications where a model is just a component of that application, and it shouldn't be seen as the only source of AI power. So I agree with that. That's a danger also. We have been switching models in the sense that we have used different versions of Claude as Claude has evolved over the past year. But we haven't moved beyond that. The design that we have would allow that to be is a straightforward approach, basically, where basically you change a couple of lines of code. We are not relying on those models to perform all the pre-processing, all the filtering, all the interpretation part. And so that's something we're trying to, isolate in large part because of your concern. We shouldn't be designing models that are depending on a particular provider.
Okay, thanks so much. That then I think brings us to the end of this session. I can't see any more questions outstanding. Just goes for me to say thank you so much, Francisco, for joining us today. That was a really fascinating talk. And for those of you who've got more interest in the topic, please do check out the Genomic AI Network's LinkedIn page and website, where you'll find lots of information about the work that we're doing, touched on a lot of the themes from today. And obviously this particular kind of forum will be now running, I think, on a monthly basis. So please do join us again next time, and we'll have a different guest speaker and a different set of discussion topics. But thank you so much for joining today, and enjoy the rest of your day. Thanks.
Thank you all. Bye.
Great session. Thank you all.
Thank you. Pleasure.